Well, good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled Single Cell Cloning: What Could Possibly Go Wrong? aka Latest Industry Thinking on Clonality. My name is Samaya, and it's my pleasure to be your Xtalks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending me a message using this chat panel. So we also have a white paper for the speaker from the speakers, and that handout is about the assurance of clonality. So audience members, if you want to download that handout, you can do so through the handouts tab in your control panel. At this time, all participants are in listen-only mode. Please also note that this webinar will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Advanced Instruments who developed the content for this presentation. Advanced Instruments is a global company offering a novel portfolio of analytical tools, including Osmotech, a robust line of microosmometers to support bioprocessing and quality control, and Solentum, a portfolio of best-in-class imaging and single-cell deposition technologies for cell line development workflows, and assurance of clonality for regulatory bodies. Now it's my pleasure to introduce our speakers for today's event, and our first speaker is Arna Andrews. Arna Andrews is currently working as a consultant to the life sciences sector, specializing in cell line development and biotechnology. Arna previously worked for CSL Limited, one of the world's largest specialty biotech companies, where from 2007 to 2019, she held the position of Director of Cell Line Development. In this role, she was responsible for establishing and managing the global cell line development function at CSL Limited CSL Behring. Now it's my pleasure to introduce our speaker, Paul. Paul Butler has worked across functions in the life science industry, including sales, field application support, and product commercialization, with experience across a range of technologies, including automation, imaging, so that's cellular, multispectral, and in vivo, label-free detection technologies, and next generation sequencing. Currently, he's working on the launch of several new Solentum products that will enable more consistency across the cell line development process and ultimately earlier patient access for novel therapeutics. So now, without further ado, I'd like to pass control to our speakers. So whenever you're ready, you can go ahead and get started.
Hello everyone, my name is Anna Andrews, and I'd like to thank Advanced Instruments, in particular Paul Butler, for giving me the opportunity to speak with you today. The title of my presentation is Single Cell Cloning, Theory versus Reality. Okay, so the things that I'll cover in my presentation today include why we need to perform a cloning step for producting cell lines, some historical perspectives that have defined approaches to cell line cloning, how during my time at CSL we used imaging technology to better understand the limitations of historical methods, some tips and tricks based on real-life experiences, and then I'll finish with a summary and some takeaways that I hope you find useful. The incorporation of a cloning step as part of the development of a production cell line is defined by ICH guideline Q5D, which states that for recombinant products, the cell substrate is the transfected cell containing the desired sequences which has been cloned from a single progenitor cell. The common approaches to meet this cloning requirement have been 2 rounds of limiting dilution cloning, or a single round of limiting dilution cloning supported by whole cell imaging, and more recently, cloning using single cell dispensing combined with imaging. Traditional limiting dilution cloning as a means to generate cell lines with a high theoretical probability of monoclonality has generally relied upon a statistical approach defined in a paper by Collar and Collar published in 1983. This approach relies on a number of key assumptions. Firstly, that cells are distributed according to a Poisson distribution. And secondly, that all cells have an equal chance of survival and growth, regardless of the number of cells seeded per well. However, Collar and Collar use limiting dilution of beads to determine their statistics. As we all know, cells do not behave like beads. The reality is that outgrowth of cells seeded at low densities is never 100%, particularly when using chemically defined animal component-free media. In addition, outgrowth efficiency is impacted by many things, such as the host cell line itself, the product it is expressing, the condition of the cells, and their environment. The advent of imaging technology has allowed us to interrogate the validity and potential limitations of this traditional approach. During my time leading the cell line development function at CSL, we used whole well imaging to look at the outcomes from traditional limiting dilution experiments. With respect to how cells are distributed when seeded at a concentration of 0.5 cells per well, we imaged 3 hours post-seeding and observed that in fact cells were actually distributed in line with the theoretical Poisson distribution. As you can see by the table, the real and theoretical outcomes are very similar. However, when we looked at outgrowth, it was a different story. This table combines the data from 21 different cell lines across 3 different projects and thus the expression of 3 different products. The way we were doing our cell line development at this time meant that each of the cell lines being cloned were in fact potentially clonal anyway, i.e. they were not bulk pools or even mini pools. As you can see, the outcomes in each case are very different. Cells expressing product 1 were highly variable in their outgrowth efficiency. In some cases, there were large numbers of wells that grew following a standard limiting dilution cloning. In other cases, there were very few or close to none. This is despite the knowledge from the imaging data shown in the previous slide, where we have some confidence cells are indeed deposited in line with the expected Poisson distribution. Cells expressing product 3 more readily grew following cloning, but in no case did the observed outgrowth match that expected if all cells deposited were to fully expand. In summary, the ability to interrogate a traditional limiting dilution cloning with the use of high-resolution imaging has revealed that there is a very large variation in outgrowth efficiency from experiment to experiment. That outgrowth never matched that expected if cell survival was actually 100%. We also observed that there is a very strong bias towards outgrowth in wells where more than one cell was actually deposited. Importantly, lines where the traditional collar and collar statistical approach would suggest a greater than 99% probability of monoclonality can be shown to often have arisen from wells deposited with more than one cell. So if we as scientists, and by extension the regulatory authorities, now know that historical approaches to cloning have significant limitations, what options do we have to improve clonality assurance and efficiency? The first and probably most accepted approach is to incorporate imaging to provide regulatory bodies with evidence of clonality, by which I mean the evidence that cell lines were derived from a single-cell progenitor. To this can be added the use of single-cell dispensing technologies to increase the efficiency of the cloning process through the precision of being able to deposit a single cell in the vast majority of wells. So, from a practical point of view, what have we learned through using imaging as an integral part of the cloning process? The first and most critical thing is that imaging at multiple time points is absolutely essential. At CSL, our approach was to image at T-1 hour after adding media to the wells. This allows for identification of any plate imperfections. We then imaged again at 2 to 3 hours following addition of the cells. This allows time for cells to settle into the well and into the focal plane. At this point, we want to find wells with just a single cell. We image again at 24 and 72 hours to track the cell and look for cell division. Then again at weekly intervals to confirm the formation of a single discrete colony with the expected growth rate. In addition to taking the appropriate number of images, it is important to define your clonality criteria. In order to designate a line clonal, we required this line to meet a number of requirements. The first was the need for a clean well at the T-1 image, followed by clean and well-focused images at further time points. We needed to see a single cell at 3 hours, either 1 or 2 cells at 24 hours, and if there were 2 cells, these needed to be in close proximity to each other. Ultimately, we needed cells to form as a discrete colony with growth rate in an expected range. And finally, cells had to be designated clonal using these criteria by 2 independent assessors. The following is an example of what we observe in a line designated clonal. We see a single cell at 3 hours, 2 cells next to each other at 24 hours, and a number of cells at 3 days. The second example is a non-clonal line, which, despite only having 1 cell observed at the 3-hour time point, actually has 3 cells at 24 hours. A single cell up here and 2 cells, or potentially even 3 cells, down in this part of the well. And these results in the formation of 2 distinct colonies at day 3. So this line would have been designated non-clonal despite only observing a single cell at 3 hours. Initially, the regulatory authorities were somewhat reticent to blindly accept the benefits of imaging as evidence to support the clonality requirements of a production cell line, and they were asking for supporting data. To this end, at CSL, we designed an imaging validation experiment to confirm our expectations and provide a statistical package to support future submission. This validation experiment was performed prior to the lab obtaining a single-cell dispenser instrument. In this experiment, we mixed 2 cell lines, one expressing a specific recombinant protein the other a recombinant monoclonal antibody. The two lines were selected as having similar growth rates, but with the ability to differentiate between them via specific assays. The cells were cultured together for a number of days and then cloned using standard limiting dilution. Each 96-well plate was imaged across 2 weeks using our normal imaging protocols. All wells from 96-well plates that showed growth were then expanded to 24-well plates, and once wells were confluent, the cell culture supernatant were assayed for both protein molecules. Based on the images, the number of cells deposited at the 3-hour time point was determined, and a determination of monoclonality made using the normal acceptance criteria that we were using. This assessment was done independently and blinded from the protein screening analysis. Putting the two outcomes together, we were able to assess the accuracy of the use of the images to determine clonality. So what were the findings? Pleasingly, across more than 500 wells that had growth, we did not observe any events where cells that were designated as clonally derived were found to express both proteins. Based on the data, our statistics group were able to derive a statistical confidence interval for clonality assurance when using our imaging protocols and analysis as basis for the decision. However, using this traditional limiting dilution cloning approach that was performed in this case, it required hundreds and hundreds of plates across multiple repeated experiments and was thus very time and labour intensive. Fortunately, the addition of single-cell dispensing technologies enables a far more efficient cloning activity to be performed. So whilst images and single-cell dispensing instruments can significantly improve the cloning process, our experience suggests that there are a number of additional things worth considering in order to maximise their value and usage. Firstly, understand as best as possible your particular cell line. What is the best media and culture conditions to support growth from a single cell? Look at the 96 well plates you choose. Assess them in terms of their image quality, cell movement, i.e. how much the cells move post-dispensing, and the cell survival and outgrowth potential. You should also consider your starting population in respect of the design of your cloning experiments. Are you starting with a bulk pool where you have a very heterogeneous cell population and your best cell lines are there in relatively low numbers and thus require potentially many plates in your cloning experiment? This is reduced if you're cloning from a mini pool where you can isolate the higher producers more readily, or do you clone from what we at CSL used to term transfectants, which may have arisen from just 1 or 2 transfection events. Lastly, I would strongly encourage you to explore the use of growth enhancers such as Instagrow, which we found to significantly improve outgrowth post single cell dispensing. In summary, what I hope you've been able to take away from today is that imaging technology has enabled deficiencies with traditional limiting dilution approaches to be investigated and highlighted. If performed appropriately, a single round of cloning associated with imaging significantly outperforms 2 rounds of limiting dilution cloning assessed using collar and collar statistics. However, imaging at multiple time points is absolutely essential, as is defining fixed clonality criteria prior to beginning your cloning experiment. Finally, the addition of single-cell dispensing technologies can significantly improve the efficiency of cloning activities and thus reduce resource requirements. So thank you for your attention today. I'd like to thank the cell line development team who worked with me at CSL in Melbourne and who were responsible for generating the data that I have discussed today. In particular, I'd like to thank Victor Turnbull, who was ably supported by Sally Ash, Rebecca De Silva, and Pia Dam.
Thank you, Arna, for that great presentation. My name is Paul Butler, and I'm a Global Product Manager for the Celentum Cell Line Development portfolio of products. I'm going to talk about the Celentum portfolio and how it aligns with the advice and tips that you heard from Arna in the previous presentation, and in the context of the latest industry thinking on clonality. So let's start where Arna left off. You might remember Arna's conclusions from her presentation. If performed appropriately, a single round of cloning associated with imaging outperforms 2 rounds of limiting dilution cloning, and that imaging should be performed at multiple time points. She also concluded that single-cell dispensing technologies can significantly improve the efficiency of cloning activities. So you won't be surprised to learn that Arna's conclusions align with the FDA and their views on the application of imaging technology for clonal assurance. The paper on the right was released at the end of 2019 and describes the FDA view on clonality and the use of imaging. A quote taken straight from that publication is that imaging technology offers an attractive way of providing supportive data to assure clonal derivation of production cell lines in lieu of additional laboratory work. And they go on to say that, in addition, these imaging systems present other non-optical based considerations as well, such as the probability that the cell is on the bottom of the well and the probability of locating the cell within the well during imaging. You additionally see a slide from another FDA presentation on the screen, and one of the comments here is that images of the entire well are needed. So, bearing that all in mind, what part does Celentum play in assuring clonality in line with the conclusions of Arna and the FDA thinking in this regard? Celentum is unique in that we cover the entire cell line development workflow and offer solutions across the entire process. At the host revival and cell culture part of the process and during transfection recovery, we offer the Insti-Thaw and Insti-Shake reagents, which improve the performance of the cell cultures during those parts of the process and help the cells to survive and grow into colonies. At the single cell isolation stage, we have the VIPS or Verified In Situ Plate Seeding System, and this essentially automates the limiting dilution process. It gives extremely highly efficient seeding and offers proof of clonality for the single cells that are seeded onto the plate. The cell metric is an imager that provides clonal assurance and is available either as a high throughput 10 plate system or a single plate system. Beyond the cloning stage, when you want to expand your cell colonies into larger formats, heading towards shaking and fed batch, we have the ICON system, and this provides tighter and viable cell density measurements at that part of the workflow. It's unique in that it uses only very small sample volumes, both for titer and viable cell density, and this allows the shaking fed batch process to begin earlier than it normally would, because those measurements don't require so much sample, and so you don't have to wait as long to grow the culture to the point where you can afford to take higher sample volumes. Beyond that, when you're finally banking your cell lines, again, we can use insta-thaw, which ensures or gives the best possible chance of cells coming out of freezing at a later stage. The instruments are controlled by the Studious software, and Studious is a data management system covering the cell line development process. It allows very rapid and error-free analysis, and part of the reason for that is that you don't need to use any spreadsheets for the analysis part of the process. All of the data from across the cell line development process is contained within a single database, and analysis becomes very fast and secure. We're going to focus now on the VIPs and the cell metric, and explain how these help in assuring the clonal derivation of your cells. So let's talk about proof, not probability, and how the VIPs and the cell metric work together to give you that proof of clonality. The reason I say not probability is because limiting dilution is based on the theory that if you follow all the correct steps in a limiting dilution protocol, then you'll have a certain level of probability of each well containing a clonally derived cell. So how do the VIPs and cell metric work together to give you that proof? So first of all, you see the VIPs on the left-hand side and the cell metric on the right-hand side, and they are linked together such that the imaging data from the VIPs is combined with the imaging data with the cell metric to give you a double lock of assurance. So how does that work? On the left-hand side, we see a droplet image from the VIPs. And what's happening is that the VIPs dispenses a small droplet in a small area of a microplate, and it then dispenses the cell into that droplet area, that smaller droplet area. It then uses high resolution imaging with high contrast, combined with an AI-based algorithm to very accurately identify whether a cell has been deposited in the bottom of the well. And the focus button that you see in this image is because you're imaging in the Z plane to create essentially a three-dimensional image of the cell to help you to prove that it is indeed a cell and not a piece of debris. So that data is then combined with the data from the cell metric. What you see in the image on the right-hand side is a cell inside a microwell, and it gives you an idea of the level of resolution and contrast that the cell metric has. So the cell metric generates an image on day 0, soon after the VIPS has deposited the cell into the well, and then it tracks and takes images daily at intervals defined by the user. And this image data can then be used to prove whether a cell is indeed a single cell or not. And those two pieces of data together provide a double lock of assurance that a well contains a clonally derived cell. So I wanted to talk about another aspect of the VIPs specifically that helps you to get away from a known limitation of limiting dilution. If you use automated cell seeding and imaging, you will know immediately about whether you've prepared the cell suspension for seeding in the first place. In the bottom left, you see one of the pages from the VIPS software, and what this is showing you is that in a particular 96 well plate, you can see green wells, which are wells that were confirmed as having a single cell deposited, and you also see red wells where we know that more than one cell was deposited and thus should be excluded. There's also an example of a grey well here, and that shows you that nothing was dispensed into that well at all. So bearing in mind that display, let's go back to the problem that we talked about before of an inaccurately prepared cell suspension, and let's see what would happen if we were doing that in a limiting dilution experiment. So let's think about what happens when the cell count is wrong, and we'll use the example of a round of cloning that should have been at 0.5 cells per well. So let's imagine a scenario where instead of preparing a 0.5 cells per well suspension, in actual fact we mistakenly have a 0.05 cells per well suspension. What you see here is that after 14 days, we realise that actually we have 0 colonies. So the plate diagram on the top is basically representing the fact that no colonies have been generated. If we had used the VIPs to do that, then at day 0, immediately after seeding, we would see this display here, which contains all wells shown in grey. And what that means is that the VIPS has realised that no cells have been deposited at all across the plate. The benefit here is that we don't need to wait up to 2 weeks before we realise that no colonies have grown. On the other hand, what if we had miscalculated and put in far too many cells per well? So in this case, let's say we had made up a suspension at 5 cells per well. In this case, we're going to have lots of colonies. In fact, it could be that the entire plate contains colonies. Unfortunately, none of those colonies, or very few of them, would contain single cells. In this case, had we done the same seeding experiment on the VIPs, the VIPs would have seeded every well with more than 1 cell per well, and would have indicated that in the software. Once again, we know at day 0, and in fact time 0, that the experiment has failed, and we can simply redo cell suspension at the correct concentration and start again, and not waste up to 2 weeks or more in learning that that all of the colonies grew and we had no single cells. Now we're going to talk about the VIPs in more detail. It's a high efficiency single cell seeder instrument, and typically you can expect that around 75 or more % of the wells in the microplate are single seeded. This is much higher than in limiting dilution, and what this means is that you can start a project with a much smaller number of cloning plates, knowing that a much higher proportion of those plates will contain single cells compared to in a limiting dilution project. The efficiency comes from the fact that it does in-droplet three-dimensional imaging of the cell, and also takes a day zero whole well image of the cell in the well, and this provides a double lock assurance. The way that the software detects single cells is through an artificial intelligence neural network, and this is a reliable way of identifying what's a single cell versus what's debris and other artifacts. You can also synchronize the data from the VIPS with the Cellmetric whole well imager, again to get a double lock of assurance. Finally, we know that single cells don't like being alone, and so I'm going to hand you over to Andrea Goff, who's our business development manager for the Instay products, to show how these reagents can aid the growth of single cells into colonies.
Yes, thanks for that introduction there, Paul. Yes, so as Paul mentioned earlier on in the presentation, there's several places in the CLD workflow that can contribute to cell stress. You know, just after transfection, that's a stressful process. Lots of cells die. During the freeze-thaw cycle, cells really, you know, don't survive very well. That's why we have to freeze down in high numbers. And then also in the clone scale-up process, there's often numerous clones that can be lost, which can be very frustrating when you're trying to make your process as efficient as possible. But probably one of the worst places is actually, as Paul just mentioned, is in the single cell isolation stage. Cells just don't like being on their own. Nowhere in our body are there any cells that aren't very close to another cell, so they just decide to die if they're not with another cell. So we've established that that's an issue, and actually regardless of the seeding method, whether it's something stressful like FACS or even limiting dilution, there will always be a proportion of cells that just die. So we've established this InstaGrow single cell growth supplement range. And this is a range of supplements that really is designed to support the cell's viability when it is on its own, and that is either of CHO or HEC cells. So we have two products in the range, and they can be used with CHO or HEC cells. And then later on I have another slide that talks about iPSC cells specifically. But this InstaGrow media is risk-free in terms that, you know, it's a defined product. It's animal component free, which is essential for developing biotherapeutics. It's optically clear for imaging. So the last thing you want when you're trying to image a single cell is to add debris in the well and then have to try and justify that it's debris. I mean, there'll always be small pieces of debris, but the last thing you want is lots of it contributed by the supplement. So this supplement is designed not to add any additional debris to the media. It really does provide improved cloning efficiency, and when we say cloning efficiency, we mean single cell outgrowth. It really improves the survival because the supplement essentially makes the cells feel like they're in the presence of other cells. It's really easy to use and fully optimized, so you know, you don't have to make your own supplements. It's just as simple to add this media to your cloning media. And I've just presented a few selection of data at the bottom there, where we have tested the product with mini pools and bulk pools, and also in a range of different media. So in every scenario, when we're comparing a pool or a single cell outgrowth, the InstaGrow outperforms the standard media components, and often people use conditioned media, which does have a good benefit, but not quite as good as this defined supplement. As well as the data I showed on the previous slide, this slide and the next will show a customer data set. So we did these really simple evaluations with customers that were having difficulties and wanted to improve their single cell outgrowth. And this example is from a customer who is a service provider, a large UK-based service provider that offers CLD services to their clients. And we just conducted this short experiment with them where they used their CHOS transfected with a standard MAB, and they develop pools. So most customers just develop pools and then ultimately take them through as individual pools through the CLD process. And yeah, so for each of these pools, they single cell dispense them into multiple 96 well plates containing their standard media, their proprietary media, as well as different supplements. So they had their own cloning media, which was something that they had developed by doing a design of experiments and adding loads of different supplements to boost their basal media. They also compared the products to, or our product, sorry, to a competitor product. And for 2 of the pools, they used InstiGrow CHO, and then for 2 of the other pools, they used InstiGrow CHO Plus. So that was the experiment they did. And then they verified clonality via validation of their seeding method a while back. So they just purely looked at outgrowth and they assumed that it is clonal outgrowth because of the validation that they had done. But in conclusion, you can see the results of all the pools and how they grew. And ultimately, for the cloning media 2, which was their design of experiments where they actually did a massive experiment to determine what supplements support their single cells, this really didn't perform well for them, and it was quite a difference when they used our supplements, something that they could just simply add and get great results. Going forward, they'll probably continue with the InstiGrow CHO rather than the CHO Plus because they saw slightly improved results with that. And yeah, they prefer this system to the competitor product because they had other reasons for not wanting to use the competitor product. In this customer case study, again, it was a large UK-based company that actually provides products as well as services, and they're working on a proprietary HEC cell line, and they wanted to improve their outgrowth, their single cell outgrowth of the parental cell line to start with. And they did this by doing manual limiting dilution into 384 well plates, containing their proprietary media with either their own standard selection of supplements or this InstiGrow HEC supplement. And they also use the cell metric to verify clonality. But in short, really simple experiment that compared the 2 different supplement types, and they saw quite a significant increase in the outgrowth of the HEC single cell. So they were really pleased, and they're going to continue to introduce this into their workflow so that they have optimized the number of single cell derived colonies that they get from the various plates that they seed in their CLD process. Finally, to finish, I mentioned earlier that we don't just focus on CHO and HEC cells. We also have a iPSC matrices called MatriClone, and also a clinical version of this product that supports the single cell outgrowth of iPSC cells, because iPSC cells, unlike CHO and HEC, are actually a bit more sensitive. And this matrices is designed to support high viability of the iPSCs as they grow out from a single cell. So it's not technically a supplement, but it's a matrices that supports the cells as they divide into a colony. And what's good about it is that it maintains pluripotency. It doesn't induce differentiation of the cells in the conditions. The solution is, or the matrices, sorry, is optimized for in-solution use, and this is very or wildly different from current protocols. Quite often, most people are very used to coating their plates with some form of matrices, whether that's a recombinant or other matrices. But the beauty of this is that it avoids the need for coating plates, and I can't emphasize enough how much time saving that gives our customers. It is quite a habit to get away from, is coating plates, but once used to it, adding this matriclone in solution is so much easier for our customers. As mentioned earlier with the other reagents, is the supplements, is you don't want them to be adding any kind of debris to the well, especially if you're trying to identify a single cell. And so this product is also optically suited for imaging. Again, it's a defined animal component free matrices, and it's actually built on a single recombinant protein, which is based on laminin. Which is ideal for iPSC cells. It does not cause any blockage when it's added to the VIPs, so if you're using the VIPs to isolate your single cells, it can be added, and it really doesn't cause any issues in the seeding process. But you can use it if you're doing limiting dilution as well. That's also acceptable and just as effective. And also, as I mentioned earlier, it doesn't impact pluripotency or the stability of the iPSCs, and they don't differentiate throughout the process. And we've got some data to show that is the case. As you can see on the slide at the bottom right, there's no effect on the iPSC pluripotency when using this product in solution. And then I mentioned earlier that we also have a clinical manufacturing grade available, and this is for those people that once they've set everything up in their process development, they can move over to an almost identical, well, it is an identical product, but it has a lot more testing applicable for clinical use. So yeah, the main thing I want to emphasize is essentially that this product is used in solution and really saves people a lot of time, and emphasizes the fact that you don't need to coat plates anymore. So that's it from me, Paul, and I'll hand back over to you.
Thanks, Andrea. And having heard about the VIPS single cell seeder and the InstaGrow reagents that you can use to maximise the chances of colony growth, I'm going to return now to the cell metric and talk about that in a bit more detail. So it's a high throughput imager, and you can see the 10 position plate stacker version of the cell metric in the picture. And as we said before, it's there to provide evidence of single cell clonality. So it can provide daily imaging. It can generate a clonality report, which is a really popular feature. And over the years, we've been featured in over 100 successful IND submissions with the cell metric. And on the image on the right, you can see one of the reasons why the cell metric is so popular, and that is the high quality of the imaging. You can see in the scale that there's a scale here of 200 microns, and you can clearly see, because of the high resolution and the high contrast, that the single cell is clearly visible. So how does the cell metric software allow us to easily identify single cells in the well? So first of all, you can see that at day 0, we can see a single cell. However, for anyone that's used a microscope for identifying single cells, you'll know that it's a difficult process, and locating that needle in a haystack is not easy. The way that the cell metric overcomes this is, first of all, it takes the measurement at day 0 for the entire whole well of the plate, It then takes an image on day 1, and it takes an image on day 2, and so on until we get to, for example, day 7. And you can imagine that this process continues from, on to potentially day 14 and beyond. What we then do, and what the Cellmetric software allows us to do, is we can look at the image of a much larger colony, like we see here on day 7, and knowing the location of that colony gives us the ability to look at the day 3 image, for example, and we know it's fairly easy to spot that smaller colony because we were looking in the right place to begin with, and the position of that smaller colony then allows us to more easily find the position of, in this case, the 4 cells on day 2, and then to go back to day 1 and find 2 cells, and ultimately go back to day 0 and find the single cell. So it's a much more logical way of identifying single cells. In addition, the Cellmetric provides confluency measurements, and you can see on this representation here that we can easily identify the wells that contain colonies and concentrate on those ones. In addition, the Cellmetric has an option of a 10-position plate stacker, so that you can do the imaging in a walkaway manner in high throughput. Finally, all of this image and clonality proof data is automatically made into a clonality report, and we see an example from a typical clonality report, and this particular example shows 3 time points in the same report, and for the first time point, we're looking at the single cell in the well at day 0. We've annotated the position of the single well in this green box, and we've also identified other interfering features that we know are debris, but it could be taken for a cell had we not performed closer analysis. And then we see the same thing on the day 1 when the cell has split into 2, and then the same on the next time point when we're looking at 4 cells, and this gives all of the information that's needed to prove clonality to the regulator. Celentum's latest instrument release is the Icon, which is designed for clone selection and characterization based on critical quality attributes. In particular, it combines the measurement of titer and cell counting and viability measurements into the one instrument. And you can see the plate-based titer assay here and the slide-based viability assay here. They're very low sample volume assays, and this allows potentially measurements of titer and peak viable cell density at a much earlier stage in the expansion and shaking fed batch process. It means that instead of having to wait until a shake flask in order to measure peak viable cell density, potentially those measurements can be made much earlier in the process without significantly using up your cell culture volume for the viability measurement, as it only takes 20 microliters. Only one standard curve is needed per project compared to, for example, the octet. And it's really designed for the expansion and shaking fed batch CLD process as mentioned earlier, and it can massively streamline the process by allowing clone selection to take place at a much earlier stage in the process. And ultimately this means earlier clone selection. Next, I wanted to turn to the problems around data management within cell line development today, and to frame this in the context of clonality. What's the point in proving the clonality of a cell in a particular well if during the analysis that follows using spreadsheets, the location of the well is somehow mistaken for a different well location? This is another regulatory issue, and it's to go back a step. The reason why this is a possibility is because cell line development involves the use of many different types of measurement. So, for example, there's the octet generating titer measurements. You have the ViCell, for example, generating cell viability measurements. You also have, of course, the cell metric and the VIPs, and potentially other instruments doing similar things. The icon that we mentioned earlier, another cell counter. The problem here is that all of these instruments generate their results in different formats. The only common format that you can use is a spreadsheet type of format. But that itself leads to complexity in that you're generating data at lots of different time points on lots of different instruments. And in order to select the best possible clone, you might have many spreadsheets up on the screen of your computer at the same time. This is time consuming, but it's very prone to error. And so for that reason, we've developed a unique data management and analysis package called Studious. In order to avoid the issues mentioned in the previous slide, Solventum developed Studious, which is a dedicated data management platform for cell line development. It means that all of the data across seeding, imaging, titer, and viability across the entire workflow are basically going into a single database, which is great for data integrity and gives confidence and consistency to the regulator. Studious manages the data from VIPs, cell metric, and icon, as I mentioned before, and it also enables the tracking of individual clones all the way from the cloning plate at the point of seeding, all the way through to final selection, even up to the point where clones go into the AMBER system. Really crucially, it removes the need for spreadsheets for data analysis. All of the data is instantly placed in Studious, and what this means is that in order to do triaging and clone selection, as we'll see later, that can be performed without the use of spreadsheet comparison. So what you get is better decisions, much quicker, and with much more confidence. At the bottom, you see what's called the history tree, and the history tree is basically a graphical way of presenting the data from all three instruments. So you can see the seeding at time 0, imaging data, which can be from cell metric orbits, the generation of a clonality report, titer measurements, viability measurements, and ultimately measurement of productivity based on titer and cell viability. The Analyze page in the Studious software shows you how you can triage samples without the need for an Excel spreadsheet. The first green box that you see here shows that you can highlight the VIPs data and also the clonal tagging data from either VIPs or cell metric. By activating the boxes here, you can limit your selection of clones only to clones that have been defined as single-seeded by the VIPS instrument or tagged as clonal within the VIPS or cell metric imaging capability. Furthermore, you can also select your confluence from the cell metric or VIPS to be a certain percentage on any given day, for example, day 14. And if you have the icon, you can also specify that the titer on day 14 or another selected day is equal to or above a certain number of mg per litre. You can also put in other criteria to do with cell viability and so on. And when you export the data, you get a pick list of only the clones that meet these user-defined parameters. Furthermore, Studious allows you to track clones all the way through the process. In this example, we have 8 clones from A1 to B2, and by hovering the mouse over a particular well, in this case A1, we can see that this clone came from a source plate called 1404 in well number B5, and the arrow is pointing to the green box, which shows you that in our plate library we do indeed have a plate called 1404. The clones can, when reformatted from the 24 well plate, can then be tracked beyond that, so you can essentially see your clones moving from seeding on the VIPS all the way through to final selection. So we'd like to end this presentation by launching a brand new product from Solentum called the VIPS Pro. And the VIPS Pro is the next generation version of the VIPS that we've talked about earlier in the presentation. And it's got the same high efficiency as the previous VIPS. It has the double lock assurance of clonality, using intelligent cell detection and using artificial intelligence to identify cells as they're deposited on the well of the microplate. VIPS Pro is fully integrated with Studious and has 21 CFR Part 11 compliance features, which make it GMP friendly, and that coupled with GMP friendly consumables, namely a new reservoir and nozzle. And the reservoir is really easy to basically load and unload into the VIPS system, and alongside clinical grade reagents. So it continues to be the industry-leading seeding and imaging system. And for anyone in the audience that's involved in the cell therapy area, where I know that a lot of people are still using limiting dilution as their seeding method for iPSCs, this is a particularly interesting development for this market, because it allows GMP friendly use of a single cell seeder that will be able to take you away from limiting dilution. More about the new VIPS Pro, then we will be officially launching the new instrument at the Bioprocess International convention in Boston in a couple of weeks' time, and we'll be at booth 618. Marta Rucka, who's the global product manager for the VIPS Pro, will be giving a talk called Proof for Probability, latest technology for assurance of clonality in cell therapy manufacturing. So I hope that anyone that's attending BPI will be available to come and view the presentation. So thanks everyone for listening, and I'm now going to open up the floor for questions and answers.
Wonderful. Wonderful. Thank you, Paul, and thank you, Arna, for that fantastic presentation. So audience members, before we start, we do have poll questions for you as well. So the first poll question is on your screens. And you should be seeing that poll question on your screen now. And that question is, how do you perform single cell cloning right now? And your options are limiting dilution or VIPs, or that could be other automated fluidics devices, or that could be I am not performing single cell seeding. So audience members, you can click on the answer that suits you best. And once again, that question is, how do you perform single cell cloning right now? All right, so it seems like the majority of audience members have voted, so I'll go ahead and close the poll and share the results with you as well. And the results are as follows. So 31% of audience members voted for limiting dilution, 8% of audience members voted for VIPs, 46% of audience members voted for other automated microfluidics devices, and then 15% of audience members voted for I am not performing single cell seeding. So thank you everyone for participating. And we also have one last poll question for you. And that poll question number 2 is on your screens. So that question is, what are the main challenges with single cell cloning for you? And your options are time-consuming processes, or that could be too labor-intensive, or lack of clonality assurance. That could also be complex, time-consuming data analysis, or lack of audit trail. So once again, you can go ahead and click on the option that suits you best. And that question is, what are the main challenges with single cell cloning for you? And audience members, you can continue sending in questions for Arna and Paul. We have received a lot of interesting questions, but if there are any more questions that you have on your end, please go ahead and continue sending them using the questions tab in your control panel. So I see that most of you have voted, so I'll go ahead and close the poll and share the results with you as well. The results are as follows. So 38% of audience members voted for time-consuming processes, 28% voted for too labor-intensive, 19% lack of clonality insurance, 16% complex time-consuming data analysis, and no vote for lack of audit trail. So thank you everyone for participating. Now we can go ahead and start our Q&A. So Paul and Arna, our first question from our audience member we have here is, what are the main differences you have observed between different types of plates, and which would you recommend? Arna, just making sure that we can hear you. So I think you're muted on your end.
Sorry about that. Yeah. Video but no audio. So we did quite an extensive analysis of plates, and we found actually that there was a NUNC non-treated plate that works significantly better than many of the other plates we were using. Originally we were using a Corning plate, but we found that the NUNC plate was better because it minimised the movement of cells post dispensing. When we looked at, I think the main issue with many of the plates we tried was actually the quality of the image. There was a lot of dirty whirls that would come up when we did our T-1 seeding, imaging, sorry, and that was a big issue with the plates, and there was often batch-to-batch problems as well. So in our instance, with this particular cell line we were using, the NUNC plates were definitely the best, but I think it's something that is definitely worth people looking at and deciding what works best for them.
Great, thank you so much, Arna. And our next question from an audience member is the InstaGrow animal origin-free material?
I'll take this one. So yeah, it's animal free.
Fantastic. Thank you, Paul. And our next question is, do you need both the VIPs and the cell metric for proof of clonality, or can you use just one?
So you can use the VIPs, which is the cell seeder and imager, to get a double lock of clonality, because you have the droplet image on the VIPs, but you also have the whole well image on the VIPs. Alternatively, you can use the cell metric to get clonality assurance as well.
Great, thank you once again. And another interesting question from an audience member. That question is, if I use the InstruGrow reagent, can I request and receive the document from the manufacturer to prove that the reagent is not animal derived for FDA approval? Oh, sorry, Paul, I think we can't hear you just because you're muted on your end.
Yeah, sorry about that. The answer is yes. We do supply a certificate of analysis that gives that information.
Great, thank you, Paul. And our next question is clinical grade HEK InstruGrow available? Sorry, Paul, just making sure we can hear you again.
Sorry about that. It's not currently available, but it is something that we are looking into right now. So stay tuned on that one.
Great, thanks, Paul. And our next question is, how do you avoid cell clumping at the early stage of single cell selection?
On that one, Samaya, can you move to the next one, and I'll answer that at the end if we have time.
Sure, absolutely. Our next question is, when performing limiting dilution, how important is it to be accurate in the initial seeding density? And what is the impact if you do not? This relates to with and without imaging.
So yeah, so I think Paul alluded to that in his presentation, and while I think for standard limiting dilution, you don't need to be, 100% accurate. You do not want to be out by, more than a factor of two, really. So, ideally, you'd want your cell concentration to be at about 0.5 cells per well. If you were closer to 1 or closer to 0.25 cells per well, that is probably okay. You'll get the sufficient numbers of wells growing up, and you'll still be able to do some statistics on that. But and if you're using something like the VIPS or the cell metric, you'll still get a valid experiment. But you do not, as Paul said, you don't want to be out by a factor of 5 or 10.
Great, thank you, Arna. And our next question from an audience member. That question is regarding ICON. Does ICON measure the titer of antibodies, or is it possible to measure other recombinant products such as proteins, VLPs, viruses?
So it's mostly for human IgG at the moment, as well as cell viability. However, again, stay tuned, because the medium-term plan for the ICON is that it will be able to measure a range of modalities, not just human IgG 1 to 4.
Great, thank you so much. And our next question is, has there been comparison of clone ranking with studious selected wells in static culture versus all clones expanded to shaking culture to show a correlation?
Yes, there has actually. So I'll be very, very open and honest about this. The ranking that we get in a static cloning plate for titer is not guaranteed to be the same as the ranking that you would get once you expand into sort of shaking and fed batch, and that we know. But people still do see some value in looking at, if you like, the normalized titer, i.e. the titer in relation to the confluence value that you get from the cell metric.
Fantastic.
Generally, the comment I'd make on that is one that is generally poor in a static situation is not likely to be that great even in a shaking situation. So it's good. It's very good at trying to eliminate the lower producers, regardless of what you do.
Agreed.
Great. Great. Thank you, Paul, and thank you, Arno. And our next question again is, how do you avoid cell clumping at the early stage of single cell selection?
Hello, so I'm happy to take that one on. So obviously clumping can be an issue, especially when you're looking at single cell seeding. So the way to prevent it will depend on the cell type that you work with. So generally there is a special dissociation protocol that we use for customers that work with iPSC cells. For customers that work with HEK and CHO cells, we generally recommend passaging the cells 24 hours before starting the seeding process. Again, this is something that is cell line dependent, and usually we get our scientists, FAS, to support customers with optimizing the protocol for their cell line.
Great. Thank you, Martha. And those were all the questions we had for today. So thank you everyone for sending those questions, and thank you to our speakers for all of those answers. If you couldn't attend to your questions, not to worry. The team at Advanced Instruments may follow up with you later on as well. And if you do have further questions, you can quickly jot down the emails here from Martha, Paul, Arna, and Andrea. So go ahead, quickly jot that down, or you can also take down this QR code here for audience members. If you have more questions, more comments, please use this. So thank you again, everyone, for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your end as well. Your participation is appreciated, as it will help us improve our webinars. I also just sent you the link of today's webinar. So this will be where the recording will be uploaded. You will be sent an email about that as soon as that is uploaded. So if you do have any questions or anything concerning that, you can also reach out to the Xtalks team about the recording as well. Now, please join us in thanking our speakers today, Arna Andrews and Paul Butler, for that fantastic presentation. We hope you found it informative. Have a great day, everyone.
Thank you.